All right, on slide 32 here, the second part of chapter 10, where we'll talk about the sense of hearing right now. If you'll notice our ear, it has external, middle, and inner sections. It provides us the senses of hearing and of equilibrium. I'd hate to go without either one of them. Our external ear consists of an auricle. This is what collects the sound, which then travels down to the external acoustic medius. Good illustration here. You can see the auricle on the outside. You can see the canal, the external acoustic medius, and then it goes to the tympanic membrane or the eardrum. Then it makes its connection, as you can see there, with most of the inner ear there. I think we'll get into most of this in a few moments. One thing I do want to point out to you here is the this nerve over here. It's called the vestibulocochlear nerve. And where do you think this might go? Does it go to the tongue, to the brain? Yeah, to the brain. So if you'll notice here, we have the cochlea, which looks like a snail in there. Well, there's something in here that moves. We also have the semicircular canals there. We have a malleus and an incus. We have the stapes. So all of this makes up the mechanism that allows us to hear, and then all of that is transmitted via this vestibulocochlear nerve. Middle ear is where we're going to find the tympanic membrane. This is what we term our eardrum. It's an air-filled space, and it houses auditory ossicles. The three auditory ossicles are the malleus, the incus, and the stapes. You can see those on the penny there, or the relative size of those on the penny. Tympanic membrane vibrates the malleus. The malleus is the mallet. This in turn vibrates the incus and then to the stapes. The stapes then vibrates the fluid that's inside the oval window of the inner ear. We have these auditory ossicles that both transmit and amplify sound waves. Transmit and amplify sound waves. Let's talk about the auditory tube, also known as the eustachian tube. I think we probably heard this termed the eustachian tube more than the auditory tube, but keep in mind the descriptiveness of the auditory tube. This connects the middle ear to the throat, and it helps us to maintain equal air pressure on both sides of our eardrum. So when we go up in height, a lot of times we're swallowing, trying to equalize, in altitude I'm talking about, trying to equalize that air pressure. We have the inner ear. It's made up of the membranous labyrinth. What is a labyrinth now? It is a maze, M-A-Z-E, and it's inside this osseous labyrinth. What does osseous reference? Osseous references something that is a bony type feature. Between the two labyrinths, we have this fluid that's called the paralymph. Para meaning around, and the lymph referencing the fluid. Endolymph is inside, obviously, the membranous labyrinth. Good illustrations here again. I would reference you to pay attention to the osseous labyrinth, the membranous labyrinth. See these semicircular canals. See where the sound is transmitted from, and then where it goes. And obviously it's going to go via the vestibular nerve and the cochlear nerve. Alright, the cochlea houses the organ of hearing, while the semicircular canals function in equilibrium. within this cochlea, this organ of hearing, the oval window, you'll have to look on your illustration there, leads to the upper compartment called the scala vestibule. And the lower one, the scala tympani, leads to the round window. You have a cochlear duct between these two compartments, and it's separated from the scala vestibule by the vestibular membrane. Well, do you see the association there? Scala vestibule and the vestibular membrane. And from the scala tympani by the basilar membrane. All right, the organ of Corti, the spiral organ. It has receptors called hair cells. Well, consider all of these little hair cells. Surface area here again. These hair cells possess hairs that extend into the endolymph of the cochlear duct. Above the hair cells is a tectorial membrane. It touches the tips of the stereocilla. These vibrations in the fluid of the, or there are vibrations rather in the fluid of the inner ear that will cause the hair cells to bend, resulting in incoming or an influx of these calcium ions. Again, somewhat electrochemical. Calcium ions, charged particles of calcium. All right. This causes the release of a neurotransmitter. Oh, a nerve transmitting component? Yeah. From the vesicles that stimulate the ends of nearby sensory neurons. Good illustration there. I encourage you to go through those. You can see where we get the vibrations set up, how they're transmitted on, and how they will eventually make their way to the centers in the brain that needs to be recognized there, or to recognize the vibrations. Talk about auditory nerve pathways. Nerve fibers carry the impulses to the auditory cortices of the temporal lobes where they are interpreted. All right, our sense of equilibrium. So this is the second part of this. We talked about the sense of hearing initially, and now we're going to talk about the sense of equilibrium that our ears and the inner ears offer us. So this consists, our sense of equilibrium consists of two parts, the static and the dynamic equilibrium. The organs of the static equilibrium help to maintain the position of our head when the head and body are still, and the organs of the dynamic equilibrium help to maintain balance when the head and body suddenly move or rotate. And have you noticed that there's sometimes when we make quick moves, it takes us just a moment, an instant, for us to get our sense of sight, sense of equilibrium even back. Let's talk about static equilibrium a little more. These organs are located within the bony vestibule of the inner ear. Within the bony vestibule. Inside the utricle and the saclic. These are expansions rather of the membranous lamprey. There's a macula that has to do with the or it consists of hair cells and supporting. We're not talking about hair cells like this. We're talking about cells within our inner ear. These hair cells, these tiny microscopic features and the supporting cells lie inside this utricle and saccule. Here you can see that. We know when our head is bent forward, when our head is upright, and if you'll pay attention to these otoliths in here that you can see there in the utricle, how they move around and they give us a sense of whether we're up or down, head bent forward or head upright. The hair cells contact a gelatinous material holding these otoliths or otoliths, these bony-like features that we have in our inner ear. Gravity then causes this gelatinous material and the otoliths to shift, and it bends these hair cells, and it generates this nerve impulse. Keep in mind, electrochemical again. These impulses travel to the brain through the vestibular branch of the vestibulocochlear nerve. We talked about that vestibulocochlear nerve early on, and this will help us to indicate the position of our head. Dynamic equilibrium. All right, dynamic equilibrium. There are 3 semicircular canals detecting motion of the head. They help us in balancing the head and the body during a sudden movement. The organs of dynamic equilibrium are called the cristae ampullaris, and these are located in the ampulla of each semicircular canal of the inner ear. They are at right angles to each other. You can see these all these hairs. You can see the hair cells. You can see the supporting cells as well as the sensory nerve fibers and how they will conduct the impulse to our brain. Hair cells extend into a dome-shaped gelatinous cupula. And like I said before, when we turn our head rapidly or our body generates these impulses, the hair cells in the cupula will bend. And we have these mechanical receptors called proprioceptors. These are associated with our joints, and we have changes. The changes that's detected by our eyes also helps us to maintain our equilibrium. Okay, slide 52. We are talking about the sense of sight. We have accessory organs, specifically the lacrimal apparatus, eyelids, extrinsic muscles, and the aid of our eye in its function. Or it aids the eye in its function. The eyelid is an accessory organ. It protects the eye from foreign objects. It's made up of real thin skin, and it is lined with conjunctiva. You can see this here. So I'm going to let you examine that. One thing I do want to point out to you, just as a matter of protection, I want you to see the yellow circular shaped cells top and bottom there, anterior and posterior of the eye. Again, you should know that is adipose tissue, and it is for protection. Look at the many layers that you have protecting your eye. They're thin, but at the same time it helps us to understand a little more about the protection of the eye. The lacrimal apparatus is what's going to produce tears, and these tears will help to lubricate our eye, cleanse the eye. We have 2 small ducts that drain tears into our nasal cavity, and these tears also contain lysosome, which is an antibacterial enzyme. Again, if you see the ZYME on lyso, it's a cleansing agent, so to speak. And the enzyme, again, I ask you to reference biological catalyst. Catalyst regulates the rate at which chemical reactions take place. So this is an antibacterial enzyme. Let's talk about the extrinsic muscles in the eye. These attach to the sclera, the outside there, and they help move the eye in all directions. You can see their attachment places here. You have several muscles that control this. Oblique is on the side, inferior rectus will be behind, superior rectus atop, superior oblique. You have a superior and inferior oblique, one atop, one at the bottom. Oblique is on the side. a lateral rectus and medial rectus, as well as an inferior rectus. Notice that the lateral rectus there is cut so you can see the nerve connecting back to the eye there in the yellow. All right, eye is a fluid-filled hollow sphere. It has 3 distinct layers. Good illustration here. You can see the optic nerve there that again that connects the information that we visualize, and then that travels to the brain where it really makes the impression. You have a lot of different features here: iris, lens, the pupil, the cornea. Talks about the aqueous humor. There's a lot of information right here, so I encourage you to see all of that. Outer layer is going to be, this is a fibrous layer, and it is transparent. That would be the cornea being at the front of the eye, and the white of your eye is going to be termed the sclera, the optic nerve, and blood vessels pierce the sclera at the posterior portion of the eye. Notice that it uses that blood vessels, the optic nerve and the blood vessels pierce the sclera. So go back and examine that illustration a little more. Middle layer, this is a vascular tunic. It surrounds, includes a coat, the ciliary body, and the iris. This choroid coat is very vascular. It is pigmented, darkly pigmented, and it performs 2 functions. It has to do with nourishment of these tissues with other tissues of the eye, and to keep the inside of the eye dark. I wonder why, to keep the inside of the eye dark. Interesting. We'll find out about that. The ciliary body forms a ring around the front of the eye and contains the ciliary processes, the ciliary muscles, and suspensory ligaments. It holds the lens. The lens now, that's kind of the hard part of the front of our eye there, and it changes its shape. That causes us to be able to focus. The ability of the lens to adjust the shape to facilitate focusing is called accommodation. Read that carefully to yourself, and I think you'll do a better job of it than I've just done. All right, the iris. All right, the iris is thin. It's a thin, smooth muscle, and it adjusts the amount of light that comes into the pupil. It's a hole in its center. Iris has a circular set and a radial set of muscle fibers. A circular set that goes around, and a radial set that would cross. You have an anterior chamber that would be between the cornea and the iris. And I reference you again to look perhaps in your text or on one of these slides here to see exactly where the anterior chamber is going to be between the cornea and the iris. The posterior chamber is between the iris then and the vitreous body that houses the lens. It's made up of the anterior cavity, and this houses the aqueous humor. Aqueous humor. Not ha-ha funny, but aqueous humor, the fluid there. Good illustration. So you can go through here, see the sclera, the choroid coat, the retina, the lens, suspensory ligaments. Notice where they are and how they're going to control that lens. All right, more illustrations for you. You look at the ciliary muscles being relaxed, look at the muscles being contracted, and how it affects the shape there of the thickness of the lens. So ciliary muscles contracted makes the lens thick. Ciliary muscle fibers relaxed makes the lens thin. Look at that carefully. Aqueous humor circulates from one chamber to the other through the pupil. Okay, inner layer consists of the retina. This contains the photoreceptors, light receptors. Photo referencing light here. It has the inner tunic, covers the back side of the eye with the ciliary bodies. In the center of the retina is macula luta, with the fovea centralis in its center. The point, this will be the point of sharpest vision, the fovea centralis. It's going to be in the center of the retina. On the side or medial to the fovea centralis is the optic disc. This is where the nerve fibers leave the eye, and this is where you're going to find that blind spot. That doesn't happen too often that we can recognize that. There's always that blind spot that we can't describe. This tells you about it. The posterior cavity of the eye is filled with a vitreous humor, not aqueous humor, vitreous humor, and along with collagenous fibers forms the vitreous body. Notice the vascularity, the macula luda, fovea centralis, where the optic disc is going to be located. So if we had but one eye, more of this would be a little easier to understand. And no, most of the time we can't just shut one eye and get the idea of this. We have to perhaps experience it. So notice where the pupil is there. And if you look back in the upper right there, about 130 or so, you're going to see a little extension there in orange. I believe that has to do with the nerve. All right, light refraction. That's refraction, not reflection, refraction. They have to be bent to be focused. Light waves have to be bent to be focused. This is called refraction. Both the cornea and the lens bend light waves that are focused on the retina as the two humors come into play here. All right, we have two kinds of modified neurons make up these visual receptors. These are elongated rods and blunt-shaped cones. Rods are more sensitive to light and function in dim light, but they produce a colorless vision. You can see those here, the rods and the cones. Single sensory nerve fibers and many sensory nerve fibers. The rods are single, the cones will be more than one. Examples of them here from a microscopic perspective. Good illustration. Cones provide sharp images in bright light, enable us to see color. To see something in detail, we move our eyes so that the image falls on the fovea centralis. When you go to the eye doctor, they check all these things. That's the reason they have so many new instruments at the optical centers. And it helps us to detect and to know more about the health of our vision. The portion, will it be there, the portion of the cones decreases with the distance from the fovea centralis. So read this carefully, look in your book, and pay attention to your illustrations, and I think you'll have a little bit better understanding about it. Keep in mind there's a lot of physics, a lot of mathematics involved here as to what's going on. Visual pigments. These light-sensitive pigments in the rods are going to appear as purple, and it breaks it down into a protein called opsin and retinal, and this is all in the presence of light. Decomposition of the rhodopsin activates an enzyme. Pay attention to what an enzyme is again, and this initiates an enzyme or initiates changes in the rod cell membrane. In the cell membrane of the rod, go back and look at see what the rod is. Thus, this will generate a nerve impulse, and these nerve impulses travel away from the retina. Look at your illustrations again, and are interpreted then as vision. There are light-sensitive pigments in the cones, are also proteins. Isn't that interesting? The light-sensitive pigments in cones are also proteins. Keep in mind, proteins are made of amino acids. And there's 20-something amino acids. There are 3 sets of cones, each contains a different visual pigment. The wavelength of light. The wavelength of light. That's not something that comes from us, but that comes to us. Determines the color that's perceived from it. Each of the 3 pigments are sensitive to different wavelengths of light. The color that we perceive depends upon which sets of cones the light will stimulate. If all 3 sets are stimulated, the color is white. If none is stimulated, the color is black. So you've got the difference in black and white. And what's in between? All right. Talk about visual nerve pathways. The axons of the ganglion cells leave the eyes to form optic nerves. These fibers from the medial half of the retina crosses over in the optic chiasma. These impulses are transmitted to the thalamus, nerve pathways called optic radiations, and then to the visual cortex of the optic lobe. As you can see here, pay attention to the triangles that is brought together here, how your eye can perceive, your eyes I should say, can perceive one object that you can focus on at the same time, sees a lot on the periphery. And notice how it all concentrates back here in your visual cortex of your optic lobe. I think that is the end of Chapter 10. We'll have more information that I will post on these 2 videos. More information to come.